So far, I've described two situations that'll give rise to non-Mendelian inheritance patterns. So the first had to do with allelic relationships that are non-Mendelian, and the second had to do with the phenomenon of epistasis. So now I want to talk about a third situation, which, when it occurs, will give rise to non-Mendelian inheritance patterns. So before I get into the details of this discussion about the concept of gene linkage, I first want to point out is that your exam scheduled for a week today will cover the material through epistasis. So our last lecture, the exam will not cover any of this ensuing discussion on linkage, recombination, and gene mapping. Okay, so to begin this discussion, the first thing I want to do is give you the reading assignments. So this is material that's drawn out of chapter 5. So again, these are the pages that you should review, as well as problems at the end of the chapter that you should practice as you become more familiar with the concepts that I'll be describing. So in addition to that, in upcoming lectures, we're also going to talk about a fourth situation in which you observe non-Mendelian inheritance patterns. And this is the condition of sex linkage. Again, I've shown you the human karyotype in previous discussions, and humans are one organism that have a chromosomal difference between the sexes, between males and females. So XX versus XY. Okay, so if you have a gene that falls on either of those chromosomes, they are going to be inherited, transmitted to progeny, and the traits that are controlled by those genes on those chromosomes are going to be transmitted in a non-Mendelian fashion. So we're also going to talk about those sex chromosomes a little bit more detail in terms of their role in sex determination. So again, both of these concepts are drawn out of chapters 4 and 7 in your book. Here are both the reading assignments as well as problems you should be looking at the ends of those chapters. Then beyond that, again, this is a little bit into the future now. We're going to be talking about a concept of cytoplasmic inheritance, another non-Mendelian situation where you will observe non-Mendelian inheritance patterns. And then lastly, we're going to also talk about the concept of pedigree. So I introduced a pedigree in our last lecture, and we're going to look a little bit more detail as to how geneticists use that as a tool to predict outcome crosses and interpret genetic inheritance patterns for traits within a family. Okay. Okay, so like I said, I want to talk about this situation of gene linkage and why and how gene linkage gives rise to non-Mendelian inheritance patterns. So the concept of linkage is pretty straightforward. It's also related to another concept that I've introduced previously. We talked about meiosis, the idea of genetic recombination. So again, remember, very early on in tetrad formation during a meiotic event, within those tetrads that form, you can have physical exchange of genetic information between non-sister chromatids to give rise to new allelic associations for the genes on those chromosomes that didn't exist in either of the two original parents. So again, recombination, genetic recombination, is a way to increase genetic variability in the pool of gametes that are produced by any individual. Okay, so that's a second concept related. We'll talk more about that as we go along. And then lastly, there's a third concept that relates to both the concepts of linkage and recombination. This is what we call gene mapping. So actually, with the ability or having the ability to detect recombination frequencies between linked genes, there's actually a practical utility for that type of information. You can build gene maps based on recombination frequencies. So we're going to talk about just introducing these terms. They're all interrelated. They all are incorporated into the discussion that we're about to embark on. And And so let's first, again, maybe redefine or define these terms a little bit more specifically. So what do we mean by linkage? So again, remember, if we're looking at a heterozygous genotype, you're heterozygous for A and B. So what did we tell you about independent assortment? Independent assortment occurs when the two genes, the A and the B gene, fall on different chromosomes within the genome. That's the situation or the condition that gives rise to independent assortment. Okay, so linkage is the opposite of that. So if you're looking at two genes, the two genes are going to be located on the same chromosome, and therefore being associated with a single centromere, they will segregate together as a unit during a meiotic event. So if you have a heterozygous genotype, you're heterozygous for A and B, and the A and the B gene fall on the same chromosome, there's actually two different ways that the alleles can be associated on those chromosomes. So you can, you can have this type of association where on one chromosome in the dihybrid genotype, the two dominant alleles are found on the same chromosome. They're said to be linked. Okay, and then on the other chromosome, you would have the two recessive alleles. And again, they would be described as being linked. But more specifically, for either of these A-B combinations, the two dominants or the two recessives, we describe that as coupled or cis linkage, because you have the same type of allele, the two dominants on one chromosome and the two recessives linked on the other chromosome. Okay, alternatively, there's another arrangement for that, the linkage associations for the alleles at those two genes. And that's depicted down here at the bottom. So in this case, you have a dominant A allele linked with the recessive B, and the recessive A linked with the dominant B on the other chromosome. Okay, so again, in this case, you have a dominant allele at one gene associated or linked with a recessive gene at the other. So we call that a trans or a repulsed linkage arrangement. Okay, so this is the concept. So linkage is, as distinct from independent assortment, linkage is when you're looking at two or more genes, the genes are going to be localized on the same chromosome, just in different positions, different loci on that chromosome. Okay, but what do we also know? When you have multiple genes found on a single chromosome, you can actually change the original linkage associations for the alleles at the two genes through recombination events. So again, we talked about this concept or this idea of recombination previously. This is what occurs within the tetrad during the very early stages of prophase 1 of meiosis. So here's a tetrad consisting of sister chromatids 1 and 2, sister chromatids 3 and 4, and then together associated into this complex that we call a tetrad. And what happens in that tetrad during prophase 1 of meiosis is that you have the formation of these what are called chiasmata. So these points of overlap between non-sister chromatids. And what's happening within those chiasmata is that you have actual physical breakage and then reunion of portions of the non-sister chromatids with each other. So in other words, you can see in this case, chromatid 1 is recombining with chromatid 3. So they're actually a physical exchange of a genetic information, this portion of chromatid 1 and this portion of chromatid 3. So you create a new hybrid chromatid that didn't of the two original parents. So here's an example of a recombination event or a crossover. Sometimes it'll be referred to as a crossing over event that in this case is involving chromatids 2 and 3. So this example. So again, when the recombination is complete, you create a hybrid chromatid that didn't exist in either of the two original parents. So now it's part the original chromatid 3 and part the original chromatid 2. Okay, so what that means is, when these recombination events occur, you can actually break the linkage associations between these alleles at these two linked genes. So in other words, if you start with dominant A and dominant B on one chromosome, through a recombination event, you can create a new hybrid chromosome that has the dominant A allele linked with the little b, or the reciprocal product would be the little a with the big B allele. So you can break the original linkage associations through recombination events. If you start with cis linkage, recombination will produce trans linkage. If you start with trans linkage as the parental type, the non-recombinant type, then a recombination event will produce cis linkage, the opposite of what it was originally. So this is showing you more specifically with our example. So here's a tetrad. We've already said the type we're looking at. You're heterozygous for A and heterozygous for B. We've told you that the two genes are linked. The A and the B gene are found on the same chromosome. So now what we're looking at is a tetrad here, and we're getting a recombination event or a crossing over occurring in the interval on the chromosome between the two linked genes. And in this case, it's involving chromatid 2 and 3. So when that recombination event is complete, you create new allelic combinations, the opposite of the original parental. So the original parental was cis. The recombinant types then are going to be trans, little a, big B, big A, little b. So after that recombination event is completed, again, during early prophase 1 of meiosis, you have segregation through meiosis 1 and meiosis 2 to create the final meiotic products. And here, these would be the final meiotic products if a recombination event occurred, as depicted in this tetrad here. So the two original parental type gametes, haploid gametes, are going to be big A, big B containing, and little a, little b containing. Again, we started with cis, that's parental or non-recombinant types for those two genes. And then these are the two recombinant types. Dominant A with little b or little a with dominant B. So now a trans linkage. So again, the point is that through recombination events, you can you can change the original allelic associations for the two linked genes. Okay. So that's we've covered the terminology of linkage, we've covered the terminology of recombination and the significance of those recombination events. So what about gene mapping? So the idea of gene mapping, I'll still use this diagram here, is that depending on the difference or the distance between the two linked genes, the frequency of recombination is going to vary. So in other words, if the A and the B loci on that, linked together on that same chromosome, if they're really physically close to each other, then what is the likelihood that they will recombine, that a chiasmata will form between them within that tetrad, and that they will then produce these recombinant type gametes? It would be very low. But if the A and the B gene were really far, physically very far apart, then there's a much higher likelihood that they will recombine and a much higher likelihood of producing these recombinant type gametes. And again, remember, these gametes are responsible for producing the progeny in the next generation. Okay, so if you have a way of detecting recombination frequencies, then you can build maps. If the recombination frequency is low, you know that the two linked genes are close to each other on the chromosome. And if the recombination frequency is high, then you know that they're far apart. So again, if you have a way of detecting recombination frequencies, you can build genetic maps based on recombination frequencies. And they've been doing that for, 100 some odd years. So here's an example. This is a very early map of the tomato genome that is based on recombination frequencies. Remember, I've told you, there's a lot of work in the early 1900s to work out the genetics, the underlying genetic basis for traits in tomato. Tomato's a food crop. So again, plant breeders want to know as much as they can about the genetics of various agronomically important traits in this organism. So one of the things that they wanted to do was, again, once they understood the concept of recombination, is they wanted to build a map. And so this is one of the early maps. They have a much better map nowadays. So there's the haploid number for tomato is 12. So there's 12 chromosomes represented in this genome. Here's 1, it's the biggest. And here's 12, it's the smallest chromosome in the tomato genome. And you can see on each of those chromosomes, they have localized specific genes that are responsible for determining specific traits. So for instance, here's one that determines the shape of the tomato fruit. So either a normal round shape or an oblate shape. Here's another one that determines, this is on chromosome 2, a gene that determines flower color. Either yellow versus white appear to be the most common allelic alternatives. Here's a gene on chromosome 7 that determines fruit color. Okay, so individual genes have been localized to different chromosomes in the tomato genome. And you can see on most of these chromosomes, there's been more than one gene localized to that chromosome. So, on chromosome 1, the largest chromosome, they've got about maybe 9 different genes that they mapped to chromosome 1. And you can see that they're in a very specific order. And actually, the distance between them has been calculated by determining recombination frequencies. So just to give you an example, this gene here, this is a plant height gene. So tall versus dwarf are the allelic alternatives. It's localized, that's where the gene locus for this plant height gene is on tomato chromosome 1. And here's a second gene that determines the texture of the fruit, smooth versus peach. So it either has a smooth skin or it has a peachy texture to the skin, the pubescence of a peach. So you can see there's an interval between those two genes. So that distance has been calculated based on recombination frequencies. And I can't see what the map distance is, but we'll just say it's relatively small. So then if you look at the next gene on chromosome 1, this is the one that determines fruit shape. You can see the distance between the texture gene and the shape gene is larger than the distance between the height gene and the texture gene. So that distance is marked as being greater in this map. Recombination frequency is greater between these two genes, the texture gene and the shape gene, than between the height gene and the texture gene. So those intervals between the gene loci on the same chromosome are determined based on recombination frequencies. The smaller the distance, the lower the recombination frequency, the greater the distance, the higher the recombination frequency. So this is what you can do. You can build a map, and this map is valuable, if you're a plant breeder or a tomato geneticist, and you're trying to design a series of genetic crosses to produce a specific outcome, a particular genetic combination among the progeny. Well, this having a map available to you of the genome, where the genes are and where they reside, is very valuable in designing those experiments. So here's one other example. It's the same idea. I told you that the fruit fly was also, the early 1900s, was being developed as a model system, primarily for research geneticists in the laboratory to do basic research. So they created a map as well once they understood the concept of recombination. So again, the fruit fly has a haploid number of 4, So you see there's 4 different chromosomes that are represented in this map. They're all being size, and they have different numbers of genes that have been localized to those chromosomes. But the idea is that individual genes have been localized to individual chromosomes within the Drosophila, within the fruit fly genome. And then what you can see is that in this map is that there's a particular order of those genes on a given chromosome, and you can see the distance between any 2 adjacent genes varies in length. So again, the bigger the interval, the greater the recombination frequency, and the smaller the interval, the lower the recombination frequency. So I'll just give you an example from chromosome 2. So here's a gene that determines a wing phenotype, dumpy wings, and here's another gene, a link gene on the same chromosome that determines an eye phenotype, clot eyes. And then down here, the next gene on the chromosome is one that determines body color, so black body. So again, you can see the interval, the distance in this map between the wing gene and the eye gene is small. So they recombine very infrequently. They must be physically close to each other on that chromosome. The 2 gene loci are very close to each other. But if you look at the eye gene, that determines the eye phenotype, and the body color gene, that interval is great. It's a very large distance. So those 2 genes recombine very frequently. And so again, these maps are determined by having the ability to detect recombination frequencies. And actually, it's pretty straightforward or pretty easy to calculate, to detect and then calculate those recombination frequencies such that these types of maps can be built. Okay, so I want to sort of build up to that. So I want to go back and look at the different gametic pools that are going to be produced under different conditions. So this is the classic Mendelian situation. So in all of these examples that I'm going to be describing, we're dealing with the same genotype. So heterozygous A and heterozygous B. So the question is what is the gametic pool that individual will produce if you have independent assortment? Okay, so that we already know that. You should be, with independent assortment, the A and the B gene being on different chromosomes, through the completion of meiosis, you should produce 4 different gametic types, haploid gametic types, all with equal frequency. And so here they're listed over here. And if this individual is a parent in a cross, it's that gametic pool that determines the phenotypic segregation pattern among the progeny. It's those gametes that produce the progeny, which will then segregate phenotypically, right? So the nature of that gametic pool is important in terms of how the progeny of the next generation will segregate. So with independent assortment, this is what we know. If we're just looking at the gametic pool, you'll produce 4 different gametic types, all with equal frequency. Okay, so that's pretty straightforward. So what if now we're looking at the same genotype, but we're saying that A and B are linked, and more specifically, they're linked in cis? And let's ignore recombination for now. Okay, so if you have cis linkage, as depicted here, without recombination, what are the gametic types that individual will produce through the completion of meiosis? Well, that individual with cis linkage will produce 2 different gametic types, big A, big B, little b, in a 50-50 ratio. So with linkage, without recombination, you can see that with an individual with the same genotype will produce a gametic pool that's different from independent assortment. And therefore, if this individual was involved in a genetic cross, the progeny, under the link conditions, the progeny would segregate phenotypically different than if you had independent assortment. That's the idea that, linkage distorts Mendelian segregation ratios. Okay, alternatively, you could have translinkage. And again, let's ignore recombination for now. What's the gametic pool that individual would produce at the completion of a meiotic event? Well, it'd be 50-50 of big A, little big B containing gametes. So this gametic pool with translinkage is different from this gametic pool with cislinkage, and both of them are different from the gametic pool of a dihybrid individual with independent assortment. So in all instances, all three of these individuals, these three different conditions that I've described, they produce different gametic pools, and if those individuals are used in genetic crosses, the progeny will segregate differently in all three cases because those gametic pools are different from each other. Okay, so let's look at the situation where now you have linkage and recombination. I should say without recombination, if you have linkage without recombination, which you will have if the two genes are linked on the same chromosome and are located physically very close to each other. So they almost never recombine. There's little physical distance between the two gene loci of the two linked genes on the one chromosome. So that's what we call absolute linkage. They never recombine. So now let's look at linkage with recombination. So it's the same dihybrid genotype. You're heterozygous for A and heterozygous for B. And here we're stating that we have cis linkage. Okay, so this is what the genotype looks like. So what's going to happen? So now we're saying that the distance between A and B are great enough that during a meiotic event they can recombine. Okay, so what's the gametic pool that this individual will produce? Well, in terms of gametic types, you produce the same four gametic types as you do with independent assortment. Big A, big B, big A, little a, big B, little a, little b. That's the same, that I showed you here. But here we saw a ratio of 1 to 1 to 1 to 1. 25% were of each of the four types. But over here, that would not be the case. You produce the same four gametic types, but they're not produced with equal frequency. The recombinant types are going to be produced less frequently. So what does that mean? Again, the parental non-recombinant types are, remember, cis linkage, so it's big A, big B, and little a, little b. The frequency of those gametic types will be 1/2 minus x over 2, where x is equal to the recombination frequency. And again, we use x because recombination frequencies can vary. The distance between any two linked genes can vary. It can be, it's variable. So we use x. Okay, so what about the recombinant type gametes? Remember, the recombinant type gametes are going to be trans in their linkage associations for the alleles of the two genes. So now big A, little b, and little a, big B, these are the recombinant type gametes, and their frequency is going to be x over 2. Again, where x is equal to the recombination frequency. So again, the bottom line is that this gametic pool here, linkage with recombination, is different from this gametic pool, different from this gametic pool, and different from this gametic pool. So again, the progeny of a cross where one of the parents has linked genes but they're recombining will segregate differently than these other three cases. And then the last example is if you start with translinkage, dihybrid genotype, the parental non-recombinant associations for the alleles at A and B are in trans. What's the gametic pool that this individual will produce? Well, now the trans associations are the parental non-recombinant gametes, these two here. So their frequency is going to be 1/2 minus x over 2. And the recombinant type gametes are going to exhibit cis allelic associations for A and B. So little a, little b, big A, big B, their frequencies are going to be x over 2. So again, a completely different gametic pool in terms of frequencies of the 4 different types of gametes. So so if this individual was used in a genetic cross, they would segregate differently than any of the other previous scenarios that I've described. So the question is then, how can you design a specific genetic cross that will allow you to sort of assay or interrogate those gametic pools to determine the frequency of each of the different gametic types? Well, it's a type of cross that we've talked about before, a dihybrid test cross. So if you take a doubly heterozygous individual, a dihybrid genotype, and you cross to an individual who's homozygous recessive, the phenotypic segregation ratio among the progeny will be a direct reflection of the nature of this gametic pool. Here, the 5 different scenarios that I've described. So with independent assortment, again, the dihybrid individual will produce 4 different gametic types with equal frequency. The homozygous recessive individual can only produce 1 gametic type. They're homozygous. All of the gametes, 100%, are going to be little a, little b containing. So therefore, the phenotypic segregation ratio among the progeny of that dihybrid cross with independent assortment is a direct reflection of the gametic pool that's produced by the dihybrid individual. And what do we know? Those are Mendelian conditions. They segregate 1-to-1 to 1-to-1. 25% of the progeny fall into each of the 4 phenotypic classes. That's a direct reflection of the gametic pool. What about with absolute linkage in cis or trans? Well, there the test cross progeny would segregate 1-to-1. So if A and B are linked in cis, absolutely, they never recombine. Half of the progeny of the dihybrid test cross would be dominant for A and B, and half of them would be recessive. That's the same direct reflection of this gametic pool. If you had trans linkage, absolute linkage, no recombination, the progeny of the dihybrid test cross would be 1/2 that are big A, little b phenotypically, and 1/2 that would be little a, big B. Again, a direct reflection of this gametic pool. So the dihybrid test cross is valuable for being able to detect recombination frequencies. So how do I know that? Well, what would you predict if you had linkage with recombination? So again, if you had started with cis linkage, the dihybrid individual had cis linkage, the progeny of the dihybrid test cross would segregate in the same frequency as what you have for the gametic pool. So you can actually calculate the value for X, the recombination frequency, by counting the number of recombinant progeny. And it would be the same thing with this situation. So again, if you have a dihybrid genotype where you have trans linkage, the progeny, the phenotypes that occur among the test cross progeny are going to segregate in exactly the same way as the frequencies of those 4 different phenotypic types are going to be the same as the frequencies of these 4 different gametes within the gametic pool. So again, all you have to do is determine the number of recombinant progeny among the test cross progeny, and that'll give you an estimation of the recombination frequency. So let me give you an example of that. So here, specifically in our example, we're going to say that we have cis linkage in the dihybrid individual, and a situation where you can get recombination. So A and B are linked on one chromosome, little a, little b on the other. You're performing a test cross. So this individual is homozygous recessive for A and B. And from that cross, this is the result. You see 4 phenotypic types, the same 4 phenotypic types that you would see with independent assortment, but they're not segregating 1-to-1 to 1-to-1. So we know that they're not independently assorting. A and B are not independently assorting. Instead, what you see is that 2 of the 4 phenotypic types, big A, big B, the 2 dominants or the 2 recessives, occur frequently. And then you have these 2 types, dominant A, recessive B, recessive A, dominant B, occurring less frequently. So those that occur, the progeny of the test cross that occur least frequently are the recombinant types. Those that occur most frequently are the parental non-recombinant types. An exact reflection of the gametic pool. So in this case, we know that there's a total of 20 recombinant progeny in a population of 100. So 20% recombinant progeny. So X is equal, the recombination frequency is 0.2 to give rise to 20% recombinant progeny in this test cross population. So 20% recombinant progeny, so X, the frequency would be 0.2, but you really don't need the frequency, you want the percent of recombination. So in our example, we've calculated a recombination percentage of 20%. And you can then use that to build a map. And the way you do that is you convert that percentage to map units, or more formally referred to as centimorgans, and it's just a 1-to-1 conversion. So here's my map now. Based on this data here from this dihybrid test cross, I've concluded that A and B are linked, and they recombine with a frequency of 0.2, so that gives me 20 map units. Gave rise to 20% recombinant progeny, so 20 map units is the interval between those 2 linked genes. So this is how those maps that I showed you previously are built. So, we'll just use the Drosophila example. What they would do is they would pick 2 of these traits, say the ones I used earlier were the wing gene and the eye phenotype gene, these 2. They would generate an F1 dihybrid for those 2 genes, and then they would perform a test cross. And they would look at how the progeny segregated. And the recombinant type progeny that resulted from the cross were very rare. So recombination was very infrequent, so you get a small interval here. But when they did a second set of crosses with clot eyes and black body, they generated an F1 dihybrid, performed the dihybrid test cross, and they recovered lots of recombinant progeny. So you can see the interval, the distance between those 2 genes is much greater. So this is how they built these maps, 2 by 2, looking at 2 genes at a time within a dihybrid test cross, and accumulate, performing lots of crosses, collecting lots of data. They were eventually able to put it all together into this one complete descriptive map of the Drosophila genome, where they localized individual genes to individual chromosomes, and they were able to determine, relative distances between the linked genes based on those recombination frequencies. So I just wanted to summarize again, what are the expectations given the different conditions? I've already gone through all of these, where we're looking at gametic pools, but then now we're looking at, how the test cross progeny would segregate. So if you perform a dihybrid test cross and you have independent assortment, your prediction would be 4 phenotypic types among the progeny in a ratio of 1-to-1 to 1-to-1. If you have absolute linkage in cis in that F1 dihybrid and you perform the test cross, you should get a 1-to-1 phenotypic segregation ratio between these 2 phenotypic types, dominant, dominant, recessive, recessive. If you have trans linkage, absolute linkage in trans, and you perform the dihybrid test cross, you should see a 1-to-1 segregation of, the dominant recessive and recessive dominant for the 2 traits. And then lastly, if you have linkage, but also recombination occurring, the genes are far enough apart that they can actually recombine, you expect to see the same 4 phenotypic types that you observe with independent assortment, but in a distorted ratio. And so here are those formulas again. The phenotypic types that occur most frequently are going to be the parental types. So that's the 1/2 minus x over 2 with cis linkage. And then the recombinant types are going to occur less frequently. That's these two here, calculated by x over 2. So again, you can count up the number of recombinant progeny, determine their relative frequency in the test cross population, and then you can use that to calculate a map distance, just like I described in the example we just went through. And then lastly, if you have translinkage, these two phenotypic classes, dominant A, recessive B, recessive A, dominant B, with translinkage would represent the parental types, parental non-recombinant phenotypes, and then the recombinant types would be cis, x over 2, here and here, big A, big B, little b. Okay? So this is how they built those maps, the maps that I showed you previously, and the same sort of maps that they assembled for many other organisms. Okay, so I'm going to stop there for now. I'll let you digest this a little bit, and then we'll continue this discussion in an upcoming lecture.